Hello students, good morning everybody. Today we are going to discuss about the locomotion and movement. So these are the two different aspects, even though they cannot be separated from each other. The movement is the movement of the individual organs in an animal, the movement of the limb, the movement of the tongue, movement of the jawbones. So all these come under the movement, whereas the organism displacement from one place to the other place comes under the locomotion. So all the animals have to move from place to place to get food material for the sake of shelter and also for the sake of breeding purpose. So here we can say that the locomotion is a type of movement, but not all the movements are locomotory movements. So simply the movement of the tongue is not locomotion. Locomotion is the displacement of the organism for different purposes, to get shelter or to get food material or for the sake of breeding purpose. Definitely the locomotion is the most important characteristic feature of the animals, except certain animals which are sedentary, belonging to phylum Porifera, and some of the animals belonging to phylum Cnidaria. Almost all the animals definitely have to move from place to place for the sake of the getting food material or whatever it may be. So as far as this locomotion is concerned, this is being performed even lower organisms also. So lower organisms like protozoans, for example amoeba, paramecium, euglena. So they have different types of the organelles like cilia, flagellae and pseudopodia. So in the different protozoans, different types of locomotory organelles are seen. In case of paramecium, the cilia are seen as locomotory organelles. In case of euglena, the flagellum is the locomotory organelle, whereas in case of amoeba, the pseudopodia are the locomotory organelles. Not only they are useful for the movement, for displacement of the organism, but they are also useful for getting food material, engulfing the food material. And even in higher organisms also, including the human being, there are certain cells like lymphocytes or macrophages or white blood cells. So they also exhibit the amoeboid movement from place to place for the sake of defensive system to protect the body against infection. So the lymphocytes also move to the site of the infection, engulf that pathogen and protect the body against infection. So even in higher organisms also, including the human beings, there are certain cells which exhibit locomotion for the sake of defensive system to protect the body against infection. So in case of higher vertebrates, including human being, the locomotion is performed by the muscles. You know that three different types of muscles are seen, like striped muscles, smooth muscles and cardiac muscles. So the striped muscles are the major type of the muscles responsible for locomotion, and of course even the movement of the different organs, whereas smooth muscles are present in the internal organs. They are also called visceral muscles, whereas cardiac muscles are found only within the heart. So three different types of muscles. Of these three different types of muscles, Striped muscles are responsible for the movement of various organs and also the locomotion of the human being. So, if you observe a striped muscle consists of a large number of muscle fibers. A striped muscle is made up of a large number of muscle fibers. So, if you observe the muscle fiber, a muscle fiber is made up of a large number of myofibrils. A muscle is made up of a number of muscle fibers. A muscle fiber is made up of myofibrils. A myofibril is made up of number of myofilaments. So, we have to know the ultrastructure of a myofilament so as to understand the muscle contraction. We know that the muscle in the vertebrate body, wherever it is present, we know that they are the striped muscles, otherwise called voluntary muscles, otherwise referred as skeletal muscles, because they are attached to the vertebral column or attached to the bones of the skeleton. So, if you observe a muscle fiber, let us suppose this is a muscle fiber. Muscle fiber is an individual cell, otherwise it is called a myocyte or sarcocyte. A muscle fiber is enclosed by means of sarcolemma. A muscle fiber or sarcocyte is enclosed by means of sarcolemma. A muscle fiber or a muscle cell is provided with a cytoplasm referred as sarcoplasm. The cytoplasm of the muscle cell is called sarcoplasm. And the muscle cell is also provided with or a striped muscle fiber or a myocyte or sarcocyte is also provided with a number of nuclei, so that a striped muscle fiber is in syncytial condition. This is the nucleus. So, number of nuclei are present within a single muscle cell or muscle fiber. Besides, the cytoplasm of the muscle cell is also provided with endoplasmic reticulum. The endoplasmic reticulum of the muscle cell is referred as sarcoplasmic reticulum. The endoplasmic reticulum is called sarcoplasmic reticulum. And again, the cytoplasm of the muscle cell is provided with all the components required for muscle contraction. It is also provided with the glycogen, and it also consists of the creatine phosphate and other requirements, and of course, number of mitochondria useful for releasing energy. So, like this is the structure of a single muscle fiber or sarcocyte or myocyte. Now, this muscle fiber is provided with a number of myofibrils. A large number of myofibrils are present like this. So, these are the myofibril. So, number of myofibrils are present longitudinally throughout the length of a single muscle cell or muscle fiber or myocyte or sarcocyte. Now, if you observe the structure of a myofibril, so this is a myofibril. the myofibril is provided with alternate dark and light bands. So, alternatively, dark and light bands are present within the myofibril. Say for example, this is the light band, which is called I band. I stands for isotropic, and again followed by A band. A stands for anisotropic or dark band. I band is light band. So, again I band, A band. So, like this, each myofibril is provided with the dark and light bands alternatively. So, this is the I band, which is dark, which is light, isotropic, and centrally it is provided with a Z line or COURESES membrane or G.P.R. So, all the I bands which are light, they are provided with Z lines. Whatever the number of the I bands, each and every I band is centrally provided with a dark line, that is called Z line. Whereas the A bands which are dark, A band is completely dark except at the middle. So, this is A band which is dark, and this A band which is dark is provided with a central light zone called H zone. This is H zone. So, again this A band is also provided with a central light zone called H zone. The rest of the A band is dark. The rest of the A band is dark, and centrally it is provided with the light zone called H zone. So, alternatively, throughout the length of the myofibril, you can come across with I band and A bands. I band is light, isotropic, with a central dark line called Z line. A band is dark, and centrally it is provided with a light zone called H zone or Hansen's node or Hansen's disc. And the part of the myofibril present between two adjacent Z membranes or Z lines is referred as sarcomere. It is called sarcomere. The part of the myofibril present between two Z lines is referred as sarcomere. And sarcomere is regarded as the unit of muscle contraction. Unit of muscle contraction. So, we know that a muscle is made up of a large number of muscle fibers. This is a muscle fiber. A muscle fiber is made up of a number of myofibrils, and a myofibril is provided with a number of sarcomeres, which are regarded as the units of muscle contraction. And that sarcomere is made up of myofilaments. Myofilaments. So, a myofibril is made up of myofilaments. Actually, they are present within the sarcomere, which is regarded as the unit of the muscle contraction. Because while we discuss about the muscle contraction, we will take the sarcomere as units, the structural and functional unit of the muscle contraction. So, sarcomere is made up of a number of myofilaments. Now, if you observe the myofilaments, so if you observe the myofilament, the arrangement of the myofilaments within the sarcomere, So, let us say these are the Z lines, because we have referred that the part of the myofibril present between two Z membranes is referred as sarcomere. So, here we are showing the sarcomere, that is the part of the myofibril present between two Z membranes horizontally, and attached to the Z membranes, there you can come across with the actin filaments. The first type of the filaments referred as actin filaments, followed by the myosin myofilament. So, like this, throughout the length of the sarcomere, the actin and myosin myofilaments are arranged like this. So, again actin filaments. Throughout the length of the sarcomere, these two types of myofilaments. So, a myofibril is made up of myofilaments, that is the unit of the muscle contraction. So, this is the sarcomere having two types of myofilaments. One is actin myofilaments, actin myofilament, and another one is myosin myofilament, myosin myofilament. So, two types of myofilaments are present within the sarcomere. Myosin myofilament is thick, whereas actin myofilaments are thin. So, if you distinguish the regions present within the sarcomere between two Z lines, the central part of the sarcomere is recognized as A band. This is A band, anisotropic or dark band. So, this is the dark band, which is having a central light zone called H zone. So, this part of the A band is regarded as H zone. This is H zone. On either side of the A band, there is a part of the I band. So, this is a part of the I band and this is also a part of the I band. So, here you can recognize this is I band. So, these two components on either side of the A band, they are the I bands. So, I band is provided with only actin filaments, whereas the A band consists of both myosin as well as actin filaments, because the actin filaments overlap the myosin myofilament at both the edges. The actin myofilaments overlap the myosin filament at the edges. So, that is why A band is provided with both actin as well as myosin myofilaments, but the H zone consists of only myosin myofilaments, where that particular part of the A band is light or H zone, which we have referred it as H zone. So, this is the structure of the sarcomere having two types of myofilaments in a relaxed condition. In a relaxed condition. So, it is the relaxed state of the sarcomere, and whenever the muscle contracts, there will be no difference between the A band and I band. Whenever a muscle contracts, so there will be no difference between the A band and I band. The entire sarcomere consists of both myosin as well as the actin filaments. So, these are the actin filaments. The entire sarcomere is made up of both actin as well as myosin myofilaments. So, here this is the contracted state. This is contracted state of the sarcomere, and these are the two Z membranes. So, there is no difference of A band and I band in the contracted state because of the sliding of the actin filaments inwards around the myosin. So, actually these actin and myosin myofilaments are thick and thin myofilaments. They are made up of proteins referred as proteins of contraction. So, the proteins which are involved, the proteins which help in contraction, two major types of proteins are involved in muscle contraction, namely myosin and actin. Myosin and actin. So, these myosin and actin myofilaments are nothing but made up of proteins like myosin protein and actin protein. So, these filaments, myofilaments are formed by those proteins. Number of myosin as well as number of actin proteins constitutes these filaments. Suppose if you observe the actin filament, the actin filament is provided with number of globular actins. Two rows of globular actins are present like this. So, this is one row of globular actin proteins. So, this is an individual protein. So, two rows of the globular proteins. So, it is globular like this because it is almost rounded. So, each globular protein will be like that. So, two rows of such globular proteins aggregate together to form this actin filament. Each actin filament is formed by two rows of globular actin proteins, and each actin protein is provided with a site, otherwise called active site, otherwise called myosin binding site. So, it is myosin binding site. So, each globular actin is provided with a myosin binding site. So, almost all these globular actins are provided with myosin binding site, and this is useful for binding of the myosin at times of contraction. Only during muscle contraction, the myosin proteins bind into the myosin binding site of actin. So, this is G actin, globular actin. G stands for globular, and the active site receives the myosin or head of the myosin during muscle contraction. So, both these two rows are provided with the active sites, and characteristically these two active sites of all the globular actin proteins are enclosed by means of another filament like protein. Another filament like protein, that is tropomyosin. So, that is a filamentous structure associated with the actin filament itself. So, this tropomyosin, which is a filament, encloses the active site or the myosin binding site of all the actin proteins. So, this is a single filament that encloses the active sites of the actin filaments, and at regular intervals the tropomyosin is provided with or attached to the tropomyosin, there you can come across with another protein that is called troponin. Troponin. The muscles which are helpful in muscle contraction are myosin and actin, whereas the tropomyosin and the troponin are called regulatory proteins. Regulatory proteins. Regulatory proteins which help in muscle contraction are tropomyosin and troponin. Tropomyosin and troponin. These are the two regulatory proteins. So, actually each actin filament is associated with two rows of globular actins, two rows of tropomyosin filaments, and also troponin at regular intervals. Now, this troponin, so let us assume that here is the tropomyosin. This is tropomyosin, TPM, tropomyosin. And attached to the tropomyosin at regular intervals, there we can come across with the troponin. So, this is the troponin. And troponin consists of three subunits, namely TNT, TNI and TNC. Troponin consists of three subunits. TNT, tropomyosin binding subunit. So, it is attached to the tropomyosin. So, it is called TNT, tropomyosin binding subunit. And the TNI, inhibitory subunit of troponin. TNC, calcium binding subunit of troponin, that binds with the calcium at times of muscle contraction. So, that is the structure of an individual actin filament. So, the actin filaments alternate with the myosin myofilament. So, the myosin and actin are the proteins of muscle contraction. Now, let us see the myosin. So, myosin which is present at the center almost are alternating with the actin filaments. Each myosin filament is made up of about 200 to 300 myosin proteins. About 200 to 300 myosin proteins constitute the entire myosin filament. And those proteins, 300 proteins, constitutes the entire myosin. Let us suppose this is the myosin filament. Each myosin protein is provided with a tail and also a flexible neck with the head. So, that is the structure of a single myosin protein. So, myosin protein is made up of a head, then this is neck and this is the tail. The head and the neck are together referred as or constitute cross bridge, and the tail comprises the main body of the myosin filament. So, from the center where you can see the presence of M line mesophragm, almost at the center of the H zone, there you can see the M line mesophragm. So, from the mesophragm or M line, these myosin proteins are arranged equally on either side. So, if there are 300 myosin proteins, 150 will be present on the right side and 150 on the left side. So, this myosin filament is mainly constituted by the tails, which is also referred as light muromycin, LMM. So, these are constituted by the tails. Equal number of the myosin proteins are present on either side of the M line, and this entire myosin filament is mainly constituted by the tails, whereas the neck and the head project outside like this. So, these are the heads of the myosin proteins. The head as well as the neck will be projected. The heads will be almost directed towards the Z line. So, you know that two Z line are present on either side of the sarcomere. So, 50% of the myosin proteins are directed towards the Z line of one side, and remaining 50% of the myosin proteins are directed to the Z membrane of the other side. So, this is how the myosin filament is constituted. So, each myosin filament is made up of about 300 myosin proteins. Each myosin protein is made up of the tail, otherwise called light muromycin, and neck and head constituting the heavy muromycin, HMM, heavy muromycin. The head of the myosin protein is regarded as motor end, which is provided with two sites, one site for binding with ATP and another site for binding with actin. This is actin binding site. As we have referred that the globular actin is provided with an active site or myosin binding site. So, this is the head of the myosin protein which binds with the actin during muscle contraction only, but not during relaxation. Again, the head of the myosin is also provided with ATP to provide energy that is required for muscle contraction. So, this is how a single myosin myofilament is constituted by means of a large number of myosin proteins. And here the neck, which is flexible, it is only because of the flexible neck the muscle contraction occurs, or the so called the actin filaments are being almost slide inwards around the myosin during muscle contraction. So, this is about the structure of a muscle fiber, myofibril, sarcomere and the protein. So, the proteins involved in the muscle contraction are myosin and actin, and regulatory proteins are tropomyosin and troponin. So, that is about the structure of the ultrastructure of the muscle fiber, sarcomere and also the proteins involved.